Hi students, you have another video from my channel which is 11th standard biomolecules. Here carbon containing any substance it undergoes the molecules. You know that. Here is that if you compare the living tissue with the non-living tissue, means what? Non-living thing, it should have the common thing is the carbon, hydrogen and oxygen. But here when you compare the living tissue with the non-living material, the living tissue should have a carbon and hydrogen are the most proportionate than the remaining non-living things. So here while we are distinguish from the non-living and living here, which both are having the same carbon compounds even though how they are differentiate. For that have to analysis the living tissue or the living matter. Analysis of living tissue. Then we came to know what are the compounds are present or the components are present in the living tissue. For that, in the analysis process, I have to take the vegetable, any vegetable or the in the animal part liver tissue or the any vegetable leaf or the any peel of any other vegetable. For that, what the required things for this analysis of the living tissue, the requirements are there which are known as the mortar and pestle. For the grinding purpose these two have to utilize. And second one is the cheesecloth or the cotton, and for it the tissue should be make it say soft. For that purpose we have to take the this is the trichloroacetic acid. Trichloroacetic acid. Trichloroacetic acid should be added. For this analysis, we should first have to collect the plant or the liver tissue, whatever it is, going to extract. This is the mortar. In this mortar, have to collect that material which we are going to analysis. If it is a liver tissue, for example, this is the pestle. With this, you have to grind it properly. With this, have to grind properly. Then after grinding, we can get the, while grinding, have to add the Cl3CCOH, trichloroacetic acid, have to add here. Then we get the slurry substance. It will become a slurry. That one should be filtered. Now have to take the one beaker. In this also one thing have to add in the apparatus. This is a beaker. In this beaker we should what we obtain here after grinding that material which is the plant or the animal means for liver. This should be filtered here through the cheesecloth or the cotton. With by using cotton or the cheese, we have to filter this material what we have taken from the mortar. This should be filtered through this cloth. This is the cheesecloth. Cheesecloth. By using this, we filter this one. Through this, we are filtering. While filtering here, we have to get the two fractions. While you are filtering, after filter this slurry material through the cheesecloth into the beaker. In the beaker, we can obtain one type of material, and over the cheesecloth we can get the other type of material. Now here, these after filtration we obtain two fractions. The first fraction which is left over the cotton or the cheese cloth, that's known as the retentant. Retentant which was left over on the paper means what? This is a cheese cloth, that's called as retentant. Here is there, this is the filtrate. Here is the filtrate and retentant. Two components we are obtained after filtering over through that cheese cloth or the cotton. Now retentant also called as acid insoluble fraction. Acid insoluble fraction. Two fractions, that is the acid insoluble fraction which is the retentant, and here filtrate which we got in this beaker, this is known as the acid soluble fraction. These two we obtained after filtration, means it's analyzing you are like this. Now here acid insoluble fraction means the retentant, which have the more molecular weight. That's the reason they should not be filtrate within these beaker, but which are have a low molecular weight, which means the less than 1000 daltons. This is the acid soluble fraction, which is the acid insoluble fraction, which is retentant, which have a more than 1000 dalton molecular weight. That's the reason it's left over in the cheese cloth. This is the way of analysis of the living tissue. Now here in this we can say here is the filtrate which is having the content we called as the biomolecules, which are present in the filtrate, and in retentant we called as the biomacromolecules. Biomacromolecules we can find in the retentant based on the molecular weight. And here we obtained these bio, micro and macromolecules. Then how can you obtain that inorganic components and inorganic elements? For that we have to take the dry weight. First you have to take the, for example, animal tissue liver, that is a wet weight. We can get in the wet weight, that should be dried, means water should be evaporate. Then we get the dry weight. This is the dry weight. The liver tissue or the plant tissue, whatever it is, no problem. That we obtain the here inorganic components and elements. How can you obtain? Means liver with the wet weight you have to take. Then it is evaporate. Dry, we will get the dry weight. Then after this should be completely bone. Then finally we get what ash, right? It means what? Carbon and water will be evaporate from the living tissue. Now we get the calcium and nitrogen, magnesium, sulfates, phosphates. These are all the inorganic components and elements from the ash. But here what we are getting now, that completely carbon, hydrogen, oxygen molecules will be there, and the proteins are there, and amino acids and lipids, all nucleic acids also will be there. These we are filtrate into the two components, as a retentant, as a filtrate. This is a difference makes these two. But anyhow, in the earth crust, what components there in our living tissue also same components are there. But percentage and aggregation of these all will be vary from the living things to the non-living things. So these known as the biomolecules, means carbon containing component called as simply biomolecules. We'll discuss the amino acids in the upcoming video. Thank you.